Hello, this is Module 2 of the Biomedical Laboratory Techniques course covering microbiology, inflammation, and infection. In this module you'll learn the preparation of media, safe culture and handling of bacteria, and identification by biochemical means and by growth patterns. At the end of this module you will also perform a histological study to identify the progressive injury induced by bacterial infection and inflammation. As we begin the semester, we are experiencing a true infectious disease pandemic, and I feel compelled to take a moment and remind you of additional measures that we have in place. As students start the lab, we will have ethanol and bleach squirt bottles available where each student will wipe down the benches to maintain sanitation. As you work through the lab, if necessary, we have hand sanitizers and sterile wipes for hands or laptops in case you have any concerns. We also will have small alcohol wipes for points of common contact such as the microscopes, where you wipe down the eyepieces before you use the microscope and after the microscope. And as each lab begins, we'll be using infrared thermometers to take your temperature to make sure that there are no students with elevated temperatures who are at risk for their lab use. Thank you very much.
The first activity in Module 2 is going to be the preparation of bacteria culture media. So bacteria can grow in liquid or on a solid agar plate, and today in lab you're going to be pouring your own agar plates. So the nutrient media that you're going to be pouring is mixed with an agar powder and water and then has been sterilized for you. So once the liquid cools and solidifies into a solid gel, bacteria can be spread on the surface, and then the bacteria will use the nutrients in the media to grow. The media has been heat sterilized and is kept in a high temperature oven. So when you're ready to pour your plates, you can use one of your safety grips and come over to the oven and take your bottle of media and then take it back to your bench to pour your plates, and one bottle of media is enough for you and your partner to pour plates. Once you've taken the media back over to your bench, you can start pouring your plates. So this is what the media looks like once it's solidified, and this is the molten media that you got from the oven that we're going to be using to pour the plates. So you can just take the cap off of the media and then take the lid off of the plate, and then you just want to carefully and slowly pour the media on one end of the plate, and then just keep pouring until the entire bottom of the plate is covered and it's about halfway full with media. Then you can just put the lid back on and let it solidify, and once the media is solidified in the plate, it should look like this. So next we're going to be taking environmental samples and letting those grow to see what bacteria comes from each sample. So you're going to get 4 plates, 2 sheet split agar plates and 2 regular plates. And you want to make sure that you label each of these plates, 2 with room temperature, because those are going to be kept at room temperature to see what growth there is under normal environmental conditions, and then 2 labeled with 37 degrees to see what increased growth there is at human body temperature. And you also want to make sure that you have your team name or team number labeled on each of the plates so you can tell which is yours. Then you're also going to be given a vial with 2 sterile swabs in it that you're going to be using to swab your environmental sample. So just take out one of the swabs and swab whatever you want your environmental sample to be. I'm going to be swabbing the inside of the biohazard bin. And after you've taken your sample, you just want to carefully streak back and forth with the swab covering the entire plate. And you want to repeat that for each of the plates. Great, and then once you've streaked each of the plates, you can go ahead and put the plates in the different buckets. So you want to put the room temperature plates in the room temperature bucket and the 37 degree plates at the 37 degree bucket. and make sure that your team name or number is on all of them as well. So next up, we're going to be doing dilution streaking, which is a commonly used procedure to find individual bacteria in a plate. So you're each going to get a plate and clinical samples. So the clinical samples are mixed media samples that are designated by the orange safety cap. So on your plate you want to write your team number and the clinical sample that you're using. And then you're going to get a pack of inoculation loops. So you want to shake out one of the loops without reaching into the bag. And then you're going to take the bigger end of the loop and dip it into your clinical sample, making sure that you actually have the sample on there. Then you're going to take your plate and across the top of one end of the plate, you're going to carefully streak back and forth in a straight line like that. Then you're going to throw away that loop and get a new loop. And then with the big end of the loop again, you're going to rotate 90 degrees from where the original first streak was. Then you're going to cross over with your first streak a little bit and then streak back and forth again in a straight line. Then you're going to throw that loop away and get a new loop. Rotate 90 degrees again, crossing over a little bit, and then just keep going back and forth. And the idea with this is to decrease the concentration of bacteria each time you get a new loop. So with your first streak, that's going to be the most concentrated, and then hopefully by this last streak that we're going to be doing, we're going to have individual bacteria. So in this last streak, we want to overlap again with the third streak we did, but be careful not to touch the first streak, and then slowly kind of just carefully carry into the middle with that last streak. And then once you're done with that, you can put the lid back on, and you want to place your plate upside down in the incubator to grow. And you want to make sure that you take your orange capped clinical sample and return it to an instructor. Another method of distributing bacteria is by using a spinning wheel and a hockey stick spreader. So you can take your plate and put it on the top of the spinning wheel like that. And then you're going to take up 200 microliters of bacterial culture media. And just as a comparison, this is what the inoculated media looks like. So you can see it's a lot more opaque, and the inoculated media is transparent. So we're going to take up 200 microliters of the inoculated media. And then just pipette it right in the center of the plate. And then you're going to take one of the L-shaped hockey stick spreaders and just place it at the center of the plate, and then spin the wheel, carefully moving the hockey stick out towards the edge to make sure that all of the bacteria has been spread evenly on the surface of the plate. So next you're going to be doing the gram staining procedure, which is a procedure used to identify bacteria based on the differences in thickness of their cell walls. So each group is going to get a gram check slide, which has 8 different sections on it, and the first 2 are labeled C+ and C-, which are pre-spotted with bacteria controls that you can look at to compare your samples to. So once you have pre-suspended your bacterial colony in media, you can come over and you're going to spot 5 microliters of your media onto one of the 6 sections. over to the hood to do the methanol fix step. You can take your slide over to the sink to begin the gram stain procedure. So you're going to take a pair of forceps and clamp in your slide, and then you're going to begin staining. So you want to open all of your bottles, so just kind of push it back so it's open like that. And this is a 4-step procedure. So first you're going to start off by covering the surface of the slide with crystal violet, and the crystal violet just stains everything on the slide, and then you're going to let it sit for a minute. Then once a minute is up, you're going to take the excess off and then slowly rinse it, and again you want to let the water drip right on the top above where you spotted the bacteria, and then angle your slide down and let the water run down the slide until the crystal violet is gone. Then you're going to slowly add the iodine, and again completely cover the surface of the slide. And the iodine is just a mordant that seals the crystal violet in. So then you're going to let that sit for a minute, and all of these stains that you're using are in order in the bottles. So then after letting that sit for a minute, you're going to dump the excess off and use the water to gently dropwise rinse off the gram iodine. And then once that is all rinsed off, we're going to use the alcohol destaining. So this one you want to be really careful and just put a few drops, and you want to do this above the white background so you can see. So just carefully add 1 or 2 drops and let it run off until there's no more crystal violet. And you don't want to overdo this step, because if you add too much of the alcohol, then it's just going to rinse off all the crystal violet, which is not fine. Alright, so then once you've done that and it's running off clear, you're going to do the last counter stain, which is going to stain the rest of the slide. So you want to, again, completely cover it for 1 minute, and then let it sit. And then after a minute, you're going to dump off the excess, and then use the water to carefully rinse off the rest of the stain. Alright, and then once that's done, you want to put your slide on the slide dryer, and then you just can leave it there until your slide is completely dry, and then you can take it back over the hood to apply the permanent cover slip. Bacteria can be identified using media supplemented with different sugars and compounds that selectively inhibit growth of gram-negative or gram-positive bacteria, or indicator dyes that change color. So 2 of the different types of selectional media that you are going to be using to identify your clinical samples are the mannitol salt and the McConkey agar. So the mannitol salt before addition of any bacteria looks like this, and then after addition and growth of gram-positive bacteria, it looks like this. And the McConkey agar before addition of any bacteria looks like that, And after the growth of gram-negative bacteria, it looks like this. So the differentiation on selective media should complement the results that you got from your gram stain procedure. Antibiotic resistance is a recurring problem that complicates effective medical treatment and recovery from infection. Resistance emerges when repeated cycles of antibiotics are prescribed to individuals or in settings where antibiotics are dispensed or administered frequently. This lab activity will examine whether any of our cultures exhibit resistance to any of several different antibiotics. The antimicrobial susceptibility test, or Kirby-Bauer disc diffusion test, is what we're going to use. And in this test, the antibiotic saturated paper discs are going to be deposited onto a petri dish that has been swabbed with a suspension of the suspect bacteria to determine the antibiotic resistance profile. So you're going to be given a bacteria and you're going to thoroughly swab that bacteria all over the large petri dish. So you just want to go back and forth in one direction, making sure to completely cover the plate, then rotate 90 degrees and go back and forth again until you have a complete just coverage of bacteria. Then after that, you're going to use the disc dispenser, and you're just going to place it on top of where your petri dish is and just push down until all the discs have been dispensed onto your petri dish, and then you can just cover it, and then you're going to wait for the bacteria to grow, and then you can use that to determine the antibiotic sensitivity profile. So once it has grown, it should look something like this, and you'll see the complete lawn of bacteria that has been grown, and around each of the discs with antibiotic on it, you're going to see a different circle range of clear where there is no bacteria, which determines how much of the bacteria was killed by the antibiotic. So some of the circles will be very large, indicating that the bacteria was very susceptible to this antibiotic and that the antibiotic killed a lot of the bacteria, and smaller circles with a much smaller diameter, which indicate that the bacteria was resistant to this antibiotic and that the antibiotic did not kill much of the bacteria. So after using a ruler to measure the diameter of each of the discs and determining the antibiotic sensitivity profile, you will have to know which antibiotic you would use to treat a patient who was infected with this type of bacteria.
During the microbiology module, having completed the activities that familiarize you with safe handling and the characterization of what the bacteria are, we now in this final activity like to turn to looking at the consequence of the bacterial interaction with host tissue. The threat of infection is everywhere, and it's continuous in the hospital. And so what we would like to do is have the students get a sense of the progression of damage to the host tissue, and also of the cell types from the host that are responding to try and counteract that injury. So what we will have you do is look at slides and describe the histology of normal tissue versus infected tissue. So as an example, what we have is lung tissue where in those open air spaces you can see the infection proceeding. So in a typical control animal, the air spaces are large, they have a very small defined bump type lining cell layer, and the gas exchanging alveoli are nice and open and free of injurious cells. This is in contrast to the lungs of animals that are infected, where now those air spaces, those cells, those bumpy cells that were lining it, have died off. They're injured, and cells are out in the open air spaces. In addition, the essential gas exchanging alveoli are no longer properly inflated. They're partially collapsed, and they're filled with the variety of these small cells that come from the host that are there trying to ward off the infection. So we can see in this slide that in certain areas there's almost complete congestion of the gas exchanging alveoli. The second way to evaluate this is to be able to sample the tissue and look at the responsive cells. So you can procedurally put a line down a person's airway and suck out or aspirate out those cells and collect those cells and examine the extent of injury and the nature of the injury. That is shown with this slide, where the cells that have been collected have been centrifuged onto a microscope slide, and we can now characterize what kind of protective cells those are. As you can see in this slide, they're fairly uniform cells that are resident protective cells called macrophages, and there are not many. We could quantify those if we had to. That's from the animal that was the control. From the infected animal now, those cells are increased substantially. Those macrophage cells are now highly enlarged in terms of their protective function. And there's a second subset of cells that have 4-part or figure 8 nuclei in them. And these are the neutrophils that come in to ingest and release factors that will kill the bacteria. So we will help you to identify these kinds of cells, whether they're healthy, normal cells in a silent state, or whether they're highly activated and functioning in protection. And then what we would ask you to do at the end of this is, given the extent of injury and given the cellular differences, to try and make your best guess, your best diagnosis of whether you think this mouse would have survived or succumbed to the infection.